Okay, so let me introduce you briefly, Andres, even if I believe all of us know you well, what you are, who you are, but just for introducing Andres to the new students we have in fifth. Andres received his PhD in biology in 1992 at the School of Science, University of Buenos Aires in Argentina, and after spending 4 years at the CONICET, the Institute for the Genetic Engineer and Molecular Biology, still in Argentina, he reached the ICGEB in Trieste, when since 2005, he holds the position of group leader in the mouse molecular genetic laboratory. This laboratory is aimed in understanding the gene functions and the relation to disease, and the development of efficient therapeutical approaches through mouse models of human disease, gene therapy, gene editing, and adeno-associated viruses. Andres' research mainly focused on neonatal hyperbilirubinemia and Crigler-Najjarr type 1 syndrome. And it's a pleasure for me introducing the presentation of Andres, entitled Severe Neonatal Hyperbilirubinemia and Crigler-Najjarr Syndrome, Lesson from Studies with Animal Models and Patients.
Okay, so thanks, Silvia, for the nice introduction. So, well, as Silvia mentioned, well, I will talk today about more or less, as I was mentioning before, what I have done in the bilirubin field since the beginning. So first, I will start sharing my screen. That is here. Okay. So the first thing I would like to do is just to thank two persons, obviously, for let's say letting me be here, and one is Tito Barale. That's well, I haven't seen him in the among the audience, but really he's the person that let me enter into research here in Trieste and gave me really support as a scientific and as a person, and really his support was fundamental for my career. And the second person is who introduced me to the bilirubin field, that is Claudio Tiribelli, that you all know. And he is the reason that I am here, because before knowing Claudio, starting collaborating with him, I knew nothing at all about bilirubin. And well, the first question that we posed with Claudio was, well, you already know all the bilirubin story, so I will be very fast. So when UGT1A1 is not present, then we have hyperbilirubinemia and potential brain damage, carnivorous, and death of the neonates. We have essentially two main causes. One is non-genetic, that is neonatal unconjugated hyperbilirubinemia, in which there is only a delay in the system to metabolize bilirubin. We are going to see this later. And the therapies are phototherapy, exchange transfusion, and the baby needs to be under phototherapy for some time. And the second one is genetic causes because of permanent mutations in the UGT1A1 gene, and here we have different types according to the residual activity of the enzyme. So the first thing, as I mentioned, was just to try to understand which were the mechanisms related to the toxification of bilirubin in the brain, because there were a series of papers and series of information related to this, in which a jaundice newborn having essentially similar levels of bilirubin have different outcomes, and there were different possibilities. As you know, there are many modifiers of the outcome of hyperbilirubinemia and the causes of hyperbilirubinemia, and among them, one of the main, let's say, players at that moment were the ADC transporters, and work from Don Ostrov, John Walshua, and Claudio postulated this hypothesis, and obviously a lot of papers that were supporting this, that both transporters at that time, they were called MDR and MRP. Now the name has changed to ABCC1, ABCB1, and they were placed in very specific region of the brain, just letting them to transport bilirubin and to extrude bilirubin from the brain to other fluids, reducing the effect of the toxic effect of bilirubin. So with the idea of trying to understand MDR and MRP, let's say, function in detoxification, at that moment, the only animal model that was available were the gun rats. But at that moment, the genetic potential, the capacity to modify the genome was not so strong for the rats. In this case, the rats are viable, they survive to adulthood, they contain a one base deletion in exon 4 of the gene, in the common region of the UGT locus, that is a very complex locus, and this generates a stop codon. So with this in mind, that the animals were alive and they had a phenotype that was not extremely severe, so they presented jaundice and cerebellar hypoplasia. Then we decided to generate an animal model by gene targeting. So we recreated exactly the same mutation that was present in the rat by introducing a one base deletion in the exon 4 by gene targeting. And obviously what homozygous animals were jaundice as expected. So this was very, very nice for us. They didn't have protein, and they didn't have also activity. The experiment of activity was done at that time by Cristina Villarosa, years ago. The animals have very high bilirubin levels, leading to cerebellar hypoplasia, a very important, let's say, histological abnormality in the cerebellum. Here you can see the Purkinje cell layers very deorganized and without the normal arborization. The cerebellum, instead of having the normal fissure, they were much reduced and also reduced in size. And the animals, as you can see here, the mutant animals, they have also problems in, let's say, they couldn't move very well compared to the wild type. And differently from what we observe in the rat, these animals, when they were untreated, they died after a few days. After 5-6 days after birth, they completely died. So since we couldn't do initially our response to our initial question regarding the role of the ABC transporter, we started to analyze other parameters, other effects. So we first transferred the mutation into another genetic background, in which we observed that the mortality was shifted. Instead of being after 5 days, it was shifted up to 11, 12 days. The animals also, they had problems. They have a kind of seizure that we didn't characterize very well, but they had also some problems. You can see that the animals are much larger because they could survive much longer. We also could modulate which was very, very important for the next set of experiments, and in general for all the experiments we have done later on. We could modulate the lifespan of the animal and the severity of the disease just by playing with the length of phototherapy. So here you can see that, for example, the red curve indicates the animals that were untreated, that they die after 12 days, more or less. Instead, if we treated the animals with phototherapy for only 8 days, they survive a bit longer. Sorry. If we treat it for even longer, up to 10 days after birth, they survive even more or less 50% survive. But if we treated the animal for 2 weeks or more, they all survive. So this was very important because this allowed us to keep the animals. And then after the cerebellar development was complete, they didn't need any more phototherapy because they survived without the addition of phototherapy, the treatment with phototherapy. So going to the mechanism, we observed that the animals have, these were in the second, the less severe genetic background. The first observation that was the activation of ER stress markers like ATF3, CHOP, CD95 that started already at P5. We also have seen the activation of inflammation markers in the cerebellum. These are studies in the cerebellum, both in astrocytes and microglia, that started essentially at P8. So after the activation of ER stress, you can see here in the images, the coloration corresponding to the activation of the different markers in the different cell compartments, and also the activation of autophagy and the triggering of DNA damage and activation of DNA damage response by the activation of or the phosphorylation of H2AX histone. And all these events occur after the initial activation of ER stress. So we think that the main driver in this animal model, at least, is ER stress, followed by inflammation and DNA damage. And since we observed that inflammation also and ER stress were a main player, so we treated the animals with an anti-inflammatory compound that is minocycline, even if it is not normally used in humans with this effect. And we observed just the survival of the animals at the higher, partial survival and partial rescue of the animals at the higher dose without modification of systemic bilirubin, partial rescue of the arborization and the, let's say, the architecture of the Purkinje cells in the cerebellum, and also a reduction in the inflammatory markers that were observed in the untreated animals. And also a reduction, even if partial, of the ER stress markers, so and apoptosis. So we can conclude that the treatment with anti-inflammatory agents could be helpful just to reduce, or at least to prevent in some manner, because these animals were treated since birth with minocycline that was given to the drinking water. So the mother drank minocycline that was then administered to the animals through the milking of through the milk of the given to the pups. So this is the main results. Then another way that we found to try to modulate or to improve the neurotoxicity is by increasing the albumin binding capacity in plasma. So this is a scheme showing the normal plasma and cerebellum, in which in a normal animal there is no bilirubin in the cerebellum, and unconjugated bilirubin is not present practically in plasma, just some minor amounts bound to albumin. In the case of the hyperbilirubinemic animal, the binding capacity is saturated, so the excess goes to lipophilic tissues such as the brain, and we have a lot of unconjugated bilirubin in the cerebellum. And the theory is that if we increase by human albumin infusion the binding capacity in plasma, the bilirubin will translocate, will migrate into plasma, and will result in a reduction of unconjugated bilirubin in the cerebellum. And this was essentially what we have observed. So with the lower concentration of bilirubin with the lower concentration of albumin infusion, we have seen a delay in mortality of the animal, but with the highest concentration and the highest frequency, we observe just partially a total rescue of mortality. Obviously, there was a very nice correlation between bilirubin levels and the amount of plasma albumin. The amount of bilirubin, the BF, we measured the free bilirubin in the animals, and it was very reduced. And also the tissue, this was BF in plasma, and the tissue bilirubin was also reduced in the forebrain and cerebellum. And going now to the original question regarding, so at that moment, at this moment of our research, we knew quite a lot of the biology or just the model, so we could address the original question. So which was the role of MDR and MRP in the detoxification of bilirubin in the brain? So for that, we obtained from Pete Borth's lab the knockout of both the animal models having the knockout of both genes, the ABCB1 MRP or ABCC1 MRP or ABCB1 and A and B, because in the mouse we have two genes for MDR, and so both genes in this case were mutated. So we crossed and we generated all the possible combinations of animals, and the first thing we observed is that in the background of hyperbilirubinemia, ABCB1 was upregulated, while ABCC1 was not upregulated in the cerebellum of the animal. So this was the first indication that MDR or ABCB1 could have an important role. Then we tested survival of the animals, the wild type, also in untreated conditions. So all the animals, as it was for the UGT1 knockout, they died in a similar manner without having any. The idea of this experiment is that if the absence of MDR or the absence of MRP may have an important role in the survival of the animal associated to the detoxification of bilirubin in the brain, so there should be a shift of the survival curve towards the left, because they could die, if it is important, they could die much faster. So this is the idea. We didn't see any difference here in these severe conditions, no difference in bilirubin levels over time. So then we tested less severe conditions by treating the animals with phototherapy from P0 to P10, in which animals survive about, let's say, mutant animals survive 50%. And what we observe here is that when the double knockout, they all die very fast. The one that was in which we had the MDR, ABCB1 that was absent, they had a similar pattern to the double knockout, suggesting that MDR1 had an important role in detoxification, because its absence resulted in the nefaster death of the double knockout animals. While the absence of MRP, they had a less pronounced effect, and this effect was not statistically significant. So we can conclude from this that MDR had a more important role than MRP and had really a role in brain detoxification. So we can conclude here from this first part that mutant mouse, the animals model we generated reproduced the main feature of neonatal hyperbilirubinemia, and the severity could modulate the severity by phototherapy. We show that the stress and inflammation are important drivers of neurotoxicity. Anti-inflammatory treatment could be beneficial, as well as the treatment, the infusion of or the increase of bilirubin binding capacity in plasma, and that MRD has a critical role in protecting the cerebellum from toxicity during neonatal development. So now we switch to the genetic disease or syndrome that is associated to unconjugated hyperbilirubinemia, that is the Crigler-Najjarr syndrome. As I mentioned, this is a genetic cause in which the gene is mutated. It's very rare, so it's less than 1 million births per less than one live birth per million of persons. Is autosomal recessive, and the only therapy for them is phototherapy that is transient, because let's say they cannot, over time the phototherapy effect is reduced due to the increase of the thickness of the skin and the change in the body weight surface ratio. And the only curative option is liver transplantation, which has a series of limitations. The liver is normal, so we started to think about doing gene therapy, and for doing gene therapy, we use adeno-associated virus. The adeno-associated virus are very small viruses. They are non-pathogenic, they are replication defective, and this is the reason of the name, because normally they were found associated to adenovirus infections. They have two genes, and they normally integrate into the genome in very specific regions. And when we remove these genes to generate our vector, instead of integrated into the genome, the virus, most of the virus remains episomal, and we replace the viral genes by our therapeutic cassette containing a tissue-specific promoter. In our case, it's a liver-specific promoter with our therapeutic cDNA, that is the UGT1A1 cDNA, and the polyase has to stabilize the mRNA. So there are different serotypes, and the one that we use is the one that has higher tropism to the liver, and then the animals or the persons are given a systemic infusion of the virus, and these end up predominantly, depending on the serotype, in our case, to the liver. We have the chance to participate into a network, European network, to generate, dedicated to creating a gene therapy for Crigler-Najjarr syndrome. And this is just, I like to show this slide, because this shows you how difficult and how complicated it is, since we are many persons working for the treatment of a few patients. So here you can see researchers, you can see clinical doctors, you can see also patient associations, also people who deals with the regulatory part, the production of the virus. So everything is very, very complicated and very long. It took us quite a long time to reach the patient. So thanks to this, we started using our animal model to test the strategy to treat later on the patient. And I will summarize here, let's say about 8 years of work in a single slide, in which we optimize the vector by eliminating the cryptic splice sites, optimizing the introns, the codons, to improve the safety and efficiency of gene transfer. We optimize the production method. We determine also the minimal effective dose that could be used to obtain a therapeutic effect in the animals and have an idea which could be the dose to use in the patients. We have we have done also integration studies, vector shedding, immunogenicity, biodistribution, and so on. And we can see here on the right, when we inject the animals, after 2 days in the animals, the mutant animal become indistinguishable from the wild type because of the production of the UDT enzyme. So after having tested all these things and showing the translatability of the approach to patients, then we started the clinical trial of the patient in patient. So this is a dose escalating phase 1/2 clinical trial. You know that normally the clinical trials are divided in phase 1 for safety, phase 2 for efficacy, and then in a larger cohort in phase 3. Due to the availability of patients that are really very few, normally in this type of rare diseases, the phase 1 and 2 are done together. This is the reason this is phase 1/2 and escalating, because we have the first cohort in which a dose of virus that is given is low, and then a second cohort of patients in which the dose is increased. And the third cohort, in these 2 cohorts, the patients are adults, so it means that they were treated with phototherapy. They are normally treated with phototherapy. This is one of the criteria. So to receive more than 6 hours per day of phototherapy to maintain bilirubin levels lower than 300 micromolar. Having no neutralizing antibodies against the virus and no fibrosis. And in the third cohort, then patients older than 10 years are included. So what we observe in the first cohort is that we have seen efficacy because bilirubin levels went down after the infusion of the virus, but then over time the bilirubin went back to basal levels. So at that time, originally there were 3 patients. Given the pattern, the results, the response of the patient with this low dose, we suspended the treatment of the patient number 3, and then we switched directly to the second cohort with a higher dose. And these are the results of patients in the second cohort, in which a higher dose was given of 5 E12 viral genomes per kilogram, in which we observed a very clear decrease of plasma bilirubin over time, and then the maintenance or a very, very, let's say, slow increase in bilirubin over time. So this was had a follow-up of 1.5 years in all cases. And the best, I think the best idea just to show how well the clinical trial work is an image of the patient, and we can see here the patient before the treatment. You can see here the yellow in the skin and the sclera of the eyes, and then the same patient after the infusion. So this worked very well, and now we are treating the third cohort of the patients with the patient older than 10 years. I don't have the data for them, but the main point of gene therapy, as I mentioned before, is that we have seen efficacy and safety of these patients, but one important point in gene therapy is to treat most of the rare diseases of the liver. They have neonatal onset, and what happened in neonatal and pediatric setting. And this is a very important concern in gene therapy, because as the liver grows, we mentioned that before, that the DNA of the virus remains as an episome, so it's not integrated into the genome. So there is a loss of viral DNA when the hepatocyte duplicates. And this is a very important concern because we cannot re-administer the AAV vector because when we have given the first administration of virus, we really, it's like a vaccination, and they produce a very high titer of neutralized antibodies against the viral capsid. So there are some alternative strategies, such as immunomodulation. We have been working also on this. Plasmapheresis. Our network has also been working on this. We have done also some work with serotype switching, but always there are some epitopes that are conserved between the different serotype of the virus. And then the other possibility is permanent genome modification. I will show my third part of my seminar today regarding the genome modification. So going back to the basics, as I mentioned before, if the virus is not integrated when the hepatocyte duplicates, most of the hepatocytes do not contain the virus, and it is lost, so there is the therapeutic effect is very low when the liver is growing, or when the liver undergoes some kind of hepatocyte proliferation due to a pathologic condition. In an adult person, this is the same story. In the case of integration, we have this orange genome integrated into the hepatocyte, and when the hepatocyte divides, the daughter cells maintain the modification, so we maintain the therapeutic effect. And this is obtained by genome editing, using nucleases or without nucleases, in which we have the advantage that using nucleases we can increase the efficiency of homologous recombination and correction of the let's say locus that we are working with. So this is what we have done originally. So we try to correct the murine locus. So actually we have two different strategies. One is to correct the mutation directly as it is here. So this is one let's say possible strategy to correct the site of the mutation in the endogenous gene, and the other possibility is to add a corrected gene into another locus of the genome that is called safe harbor locus. In the case in which we correct the mutated gene, remember that in our mouse model we have a one base deletion, so it was relatively simple to set up a strategy for this. The advantage is that we have a regulated expression of the corrected gene. The disadvantages are that the efficiency of correction is low, may not be sufficient, and as you know, diseases are the result of many different mutations, so in this case we may need mutation-specific strategies that from a point of view of translation and commercial, let's say, development is not convenient. And this is what we observed in our case. We corrected the mutation of the UGT1A1 in the exon 4 by homologous recombination using Cas9. I don't know why this appeared. And then we observed that the animals had much lower bilirubin levels than the untreated animals, but they didn't reach wild-type levels because bilirubin was still very high. So at that point, we also performed some other strategy by inserting the therapeutic cDNA into the albumin locus. In the first case, we didn't use nucleases because this region is relatively high recombinogenic, and the low efficiency of recombination is compensated by the high transcription driven by the albumin gene. So this was our first approach. We did this in collaboration with the laboratory of Mark Kay at Stanford, in which they have done the original work using the factor 9 cDNA, and they reach up to 0.5 recombination efficiency. In our case, when we use the GFP cDNA, green fluorescent protein, we obtain a recombination efficiency that was lower than what they have obtained before, but was still in a good therapeutic range, up to 0.15%. You can see here that the cells are grouped in clusters, indicating that the recombination event was very early, and the cluster of green cells is the consequence of duplication of the original event, and they are the daughter cells that still maintain the correction. Then we move to our animal model. Well, we characterized the molecular, the corrected insertion of the GFP cDNA, which this is the sequence of the chimeric mRNA. I didn't mention before, I will explain better now. So the therapeutic cDNA is inserted just upstream of the stop codon using a peptide 2A that results in ribosomal skipping. So after correct insertion of our therapeutic cDNA by homology-directed repair, then we obtain an mRNA that is a chimeric mRNA containing the full albumin open reading frame, in frame with the peptide 2A, and in frame with the therapeutic mRNA, GFP or UGT, and then the normal albumin 3' UTR. And this is translated into two separate proteins thanks to the presence of the peptide 2A that results in ribosomal skipping. So this is the basis of the approach. When we did this approach, we observed that all animals that were treated for 8 days with phototherapy to increase just the time in which the recombination event could occur. This is the idea. And we observed that all animals survived, but bilirubin levels was still not near the wild type, even if it was into the safe levels, and the recombination efficient, and well, histology was normal. I am not showing histology patterns and these things, but everything was normal, and recombination efficiency was much lower than we obtained, probably due to the longer cDNA size of the UGT compared to the factor 9 or to the GFP cDNAs. So after that, we switched. We have seen that recombination occurs, and we have to improve just the events of recombination. And for that, we started using the Cas9 enzyme that, as I mentioned you before, the generation of a double-strand break at the site of the recombination increases the frequency between, let's say, 100 and 1,000 fold. And this is what we have done. So we use the same strategy, with the difference that this time we have given in parallel, so to the animal, the Cas9 enzyme to the animal. And this is what we observe. This is without the enzyme, what I call here HR. And this is with the enzyme for the GFP. You can see here that we obtain up to 15% more or less the recombinant hepatocyte, with some hepatocyte reaching, in this case, up to 20-25%. We further improve over time this and reaching about 30% recombination with GFP. And with this strategy that we tested our animal model just by doing, as I mentioned before, the co-infection with the two viruses. The virus is containing the donor DNA and the Cas9. As before, we treated the animals with 8 days of phototherapy to let the recombination event to occur and to start to produce UGT1A1 enzymes. And then we tested the bilirubin over time. And what we observed as before is that all the animals survived, but this time bilirubin was maintained all over the duration of the experiment at levels that were identical to the level of the wild-type animal. The efficiency of recombination was much higher, so we reached about 3–4% of recombination in the hepatocytes, and the amount of protein production was even higher than the one that is produced by the wild-type animal. So we can conclude here that this approach is very effective in, let's say, in rescuing the phenotype of the animal, and we can conclude that the episomal gene therapy is less effective in the neonatal period. We can rescue completely the neonatal lethality without using site-specific endonucleases, and targeting the Cas9, we increase the efficiency of recombination, reaching very high levels, in the case of GFP up to 20%. And in the animal model with the UGT1A1 cDNA, we completely rescue the phenotype, and this is a very effective method to correct the Crigler-Najjarr syndrome, and we still think that additional experiments are needed to translate the approach to the clinic. We have done, I didn't mention, some safety studies of the Cas9 targeting in this locus, and everything looks well. So next slide, I would like to thank the people who participated in the studies. I would like to thank mainly Giulia Bortolussi, that probably most of you also know. She did most of the experiments. She developed the animal and participated in all the experiments that I have shown here, and also the other members of the group that participated: Alessia De Caneva, Simone Bonoret, Luca Bocco, and Fabiola, and Bipin, and Alessandra. So, and the new members of the lab. And I also would like to thank Claudia, as I already mentioned, Christine and Silvia, and also, let's say, to all the people that participated in this project over the years, and the founded agency that allowed us to perform these studies. So thanks to all of you for your attention, and